I should say, well, my great interests are really making glycoconjugates using enzymatic methods. But over the last 10 years, we've become very interested in analytics. In particular, given that for our sort of enzymatic reactions, we needed analytical techniques, and there didn't seem to be sort of very generic analytical techniques around, and we had a particular problems, which I'm going to be talk about, and that made us think a little bit about the fact that there is still no generic carbohydrate sequencing methods. You know, obviously, as you all know, we can sequence peptides. And proteins, we can sequence DNA, of course, but for carbohydrates, we need these very, specific sequencing methods, which we've already heard about, and they're fantastic, but they tend to be a little bit specific for natural glycoconjugates and for particular classes. So we were quite interested in developing more generic classes, and that's what I'll be talking about today. And I should say this is a collaboration with my mass spectrometry colleagues who I'm going to mention. So what has really sort of frustrated me as a chemist is that when we do sort of carbohydrate sequencing, in particular of complex glycoconjugate, we do get a sort of picture of it, but it's still not particularly sharp in chemical terms. If you look at, I'll do this. Yeah, that's fine. Sorry, I just remembered. So it's not particularly sharp in chemical terms. There are always ambiguities, and we've already seen this beautiful work today, and there are always sort of brackets, and you don't quite know on which arm the sialic acid is, and it gets worse when you don't really have a good understanding of the biosynthetic pathways. I had a very talented colleague, Claire Ayers, who was a mass spectrometrist, and we started to think about how we could develop techniques that could address this problem about 10 years ago. And what has attracted us very much is this whole shotgun sequencing approach, which is generally used in DNA and proteins. And really what you do is you don't sequence the whole glycoconjugate, you sort of fragment it. And of course, there are fantastic fragmentation methods available for nucleic acids. And then you sequence the fragments and determine the fragments with high accuracy. And then you somehow really assemble these again. And you need computational method to deconvolute it. I think the attraction about this method is not only that you can get full sequence determination, which I have to admit, in carbohydrates is challenging, but you can actually look for certain motifs. And very often, I think in carbohydrates, in glycoconjugates, we want to look for certain motifs and see whether they're there or not. And that's sort of a lot of the problems we had. We're really concerned with these motifs, because all you then had to do is fragment it and then find, that particular motif in your mixture and then really determine the sequence of that. And that's what I'll be talking about today, really looking for motifs using this fragment-based approach. And when I talk about sort of sharpening up our sequencing, I'm obviously talking about full determination of all the stereochemistries. Which hexos do you have? Which linkage do you have? What is the connectivity? Is it alpha and beta? And so on. And ideally, we want to do this de novo, not having, all this to determine all the biosynthetic pathways and so on, having this background knowledge. Now, in terms of fragmentation of carbohydrates, we unfortunately don't have restriction enzymes so much. We have some, but we have got obviously PNGAs, but as you all know, that's more difficult for other glycoconjugates. But we have got this fantastic method of fragmenting carbohydrates into fairly defined fragments, all developed by, for example, Doman and Costello. I think that's really the classic work in this area. So we know quite a lot about fragmentations of carbohydrates in the gas phase. And we're getting very defined fragments, and we're all using these fragments for structure determination. So when you use a fragment-based approach, we thought, well, can we do this all using mass spectrometry and using these sort of fragmentation methods which are already there for mass spectrometry, generating these sort of fragments. And I don't think I have to explain it very much, but just to say that we get these B and C fragments, which are fragments that are either way of the glycosidic linkage, and you can also get, we've heard about this already, these cross-ring fragmentations. But it's particularly the cleavage of the glycosidic linkages we were interested in. So that the protocol to sequence a carbohydrate in analyte would just look like this. you would have some sort of analyte. So this is maybe an unglycan structure, so this would not be necessarily labeled, could be labeled, but not necessarily. Then you do just dissociations in your mass spectrometer using various dissociation methods into fragments, and these could be smaller fragments or even monosaccharides using the sort of Doman-Costello type of fragmentation patterns. And then you could determine the mass spectrum. And of course, if you get M over Z values, that doesn't give you the full information of these fragments, but it's a very useful method. So mass spectrometry is probably, we've been hearing so much about it at this meeting, is probably the best method to deal with complexity, sensitivity, and so on. But it doesn't obviously tell us anything about the three-dimensional structure. So you can link this to HPLC and LC and so on and compare it to standards. But it would be very nice if you could link the mass spectrometry in the gas phase with other orthogonal methods. And we've already heard about ion mobility, and ion mobility is something my colleague Claire suggested 10 years ago. So we really started to look into, can we combine mass spectrometry with ion mobility spectrometry? And ion mobility tells you something about the shape. You still need standards, but they're very, very well defined measurements. And then actually we had colleagues joining us, Isabelle Compagnon and Kavin Pagel and so on, and they've now managed to do IR as well in line as a hyphenated method with mass spectrometry. So really what we want to do is get three-dimensional information on these fragments using these methods. And the reason we're particularly interested in fragmentation for this approach is that if you do ion mobility mass spectrometry on the top compound here, the ion mobility requires you to compare the measurement with a standard. It's a bit sort of like LC. I explained a little bit more about that. And so for every analyte, you need a standard at some point. So you need a huge database of standards. But if you go to the fragments, the number of standards you need is reduced. So if you just think about N-glycans, a lot of these diverse N-glycans share the same substructures. So you really reduce your need for databases, numbers of samples in the database, as you go down to smaller fragments. So the question is, how does this approach work? And I should say, in the last 10 years, I think this has been more and more accepted, and not only our group, but other groups as well have used this sort of approach of determining structures of glycoconjugates. For those of you who don't know much about ion mobility, just a quick couple of slides to explain it. So it's a gas phase technique, so it can be used in line with mass spectra. So you just isolate an ion by M over Z value. So that's also a nice separation technique for this further analysis. Then you just put it through this drift tube. You don't always get a sharp peak because what we are measuring here is really what is called the CCS value. And you can think about the CCS value as putting the ion into sort of a sphere, and the radius of the sphere is related to the CCS value. And actually, it's a very, very important reproducible physical parameter. It depends a bit on the drift gas you're using in your ion mobility tube. But of course, these are very flexible molecules. They have different conformations. So very often with these glycans, in particular as you go to larger glycans, you get fingerprints. You don't necessarily get just one peak. The instrument is off the shelf, and in fact that became available when we were starting, so that really spurred us to do this. So this is an instrument from Waters, but we've heard about another instrument a minute ago. So it's basically like your normal mass spectrometer, just that in the middle you have this ion mobility tube, and you can just integrate the ion mobility measurements with your fragmentation and M over Z value and so on. So this is already off the shelf, and as I said, Claire Ayers and Perdita Barron are my colleagues who run this instrument. Because it is sort of basically an electrophoresis chromatography technique, the resolution you get depends on the length of the tube, and obviously it's fixed in this instrument. But people have realized that this is a very useful technique and have tried to increase the resolution. So there's now another instrument on the market, a circular version, where you basically have the ions around a circle, and that obviously dramatically increases your drift tube length, and that also increases your resolution. So the data I'm showing you is on the left-hand instrument. Some of the resolution is not particularly impressive, perhaps, but it's very reproducible. But as I said, just to point to the fact that there are, better instruments now available. So as I said, we got into some applications we used. So we got into this because we were looking for a glycosyl transferase, is one of the mucin ones, core mucin ones, which normally transfers GalNAc onto a peptide. It's the T2 mucin transferase. And we found that in vitro, it was also transferring GlycNAc at different Km values, but it was also doing that. And that seemed quite an interesting finding. We wanted to publish that. And the problem was, it was very difficult to prove that we had indeed transferred GlycNAc onto these different peptide substrates which we're using. If you think about it, an analytical technique that could distinguish between GlycNAc and GalNAc is on peptides is quite challenging. You really have to get sort of into NMR or into using LC methods with standards and so on, and that's really, really challenging. The reason why we thought we needed to show this is because there's an epimerase which is very abundant, and we were wondering, we were worried that we couldn't publish this because people would argue, well, you just there's an epimerase there, and you're transferring your GalNAc and not your GlycNAc. So that's sort of, so this to realize that it's really difficult to analyze sort of structures like this, in particular if they're unnatural structures like the one we were making. But also, there are lots of glycoconjugates where the biosynthetic pathways don't help us very much. And one project I won't talk much about was on metabolites. So there are a lot of plant metabolites which have various mono, di, triglucosides in different linkages. They're incredibly difficult to analyze. And we had a very nice paper with Birger Möller, some plant biologists, using this sort of fragment-based approach where we were able to assign the structure of these plant analytes. But I won't talk very much about it today. Because this is more sort of a glycomics audience. So just to show you the data, so you can actually do ion mobility on the glycopeptides, and we were making them in vitro, by adding either UDP galactose or UDP glucose, and we could actually show by ion mobility, and you can see the top trace there, that we get different traces. The red trace is different from the blue trace, so the two peptides were different, which gives us some confidence that it was already, that these were different. But of course, we need to compare this to authentic standards. We would have had to make these chemically, which would have been very, very tedious. And that's why we started to think about a fragment-based approach. So the question is, if you fragment this peptide, so with chemical ionization, for example, so you do this in the mass spectrometer, you generate your ions, your oxonium ions here. And the question was, could you then, after fragmentation, do ion mobility and separate these two ions? And that would be obviously a much more universal method because you could use this for any glycopeptide. It's independent of the sequence. And to our surprise, the separation, as I said before, is not brilliant on this instrument for these two, but they're really, really similar. And this was extremely reproducible. And it allowed us really to take any glycopeptide that had either GalNAc or GlycNAc and tell which one was which, independent of having any glycopeptide standards. The only standard we need here is a glycoside that has either got GalNAc or GlycNAc, and it could be any of these glycosides. So really sort of a proof of principle that really helped us to sort of characterize these products. We also then, as proof of principle, applied this to N-glycans. Obviously there we know what the structures are. And just to show that, when you do this, these fragments, independent on where they come from which analyte they come, in the gas phase, they will always have the same CCS value. So, for example, if you generate fragments from this N-glycan on the top, you can either make the sort of larger fragments or the monosaccharides and compare them to fragments that come from smaller standard substructures. They line up absolutely perfectly. And, you can then do a sort of thought experiment and saying, okay, by knowing all the CCS values of these small molecular weight substructure standards, I can actually then deduce the full structure. So I only need fragment standards. As I said, emphasizing this point, you need a much smaller database of standards for this fragment-based approach. One thing we were a bit worried about is cleaving the glycosidic linkage, because surely you would actually lose your information on alpha-beta anomericity. And if you wanted to really sequence carbohydrates, you have to determine whether things are alpha or beta. Of course, mass spectrometry doesn't do that terribly well. There are some methods looking at sort of different heights of peaks, but generally mass spectrometry is not very good. So if you have alpha, the diglucoside, 1,2 diglucosides, alpha or beta, they give you the same mass spectrum. That's sort of as expected. So the question is, what about when we then combine this with ion mobility? And to our surprise, there seemed to be a really reproducible anomeric memory effect. So depending on whether the glucose ion comes from an alpha or beta linkage, you get an additional peak. So this additional peak, can you see this here, is this dark peak here. As I said, it's small but really reproducible, irrespective on what your linkage is. So we measured all the diglucosides, with all the alpha and beta ones with different regioselectivity. This pattern that you always get on the alpha glucoside, this extra peak held. So that means there's this memory effect. So you can use this method also to to determine the glycosidic linkage, which we felt was a really important step forward. I should say we've also taken the fragments which you generated and looked at them by IR, and by IR they also look very different. So there's really the alpha and the beta anomers generate different fragments under our measuring conditions. And that really means that we can really determine everything sort of de novo on glycoconjugates in principle. Obviously, we haven't shown that for every structure, but we have shown examples of several structures where this holds. So we can distinguish between galactose and glucose, between alpha and beta linkages, and of course through the cross ions, we have already seen some beautiful work there. You can distinguish what the connectivity is. And really, in principle, this method allows you to fully assign carbohydrates. But as I said, we have really used it mainly to look at particular isomeric motifs. And if I just show you quickly a few examples. So, for example, you might want to know about a certain motifs on an antibody or glycoprotein. And you're all familiar with this slide here, so I don't have to go through that. And just to show you one example, we had a project a few years ago where we wanted to develop a alpha 2,6 sialidase that was selective for alpha 2,6 linkages over 2,3 linkages. To our surprise, in the literature, there are sialidases that cleave 2,3. Sialidases cleave both 2,3 and 2,6, but 2,6 selective sialidases, hadn't been reported. And we developed this enzyme. We called it a pseudosialidase. It was actually a transferase that we used in reverse. What we hoped that this enzyme would do, even in the context of a complex glycoprotein, really only cleaves the 2,6 linkages. But we somehow had to prove that. And as you know, if you sort of helic, you can't really distinguish terribly well between alpha and beta, and you have also got a broad mixture. So what we did is we used this fragment-based approach, and in particular we used out of this forest of M over Z peaks, we isolated this B3 ion, which you can see here, which is that trisaccharide that crucially contains the 2,3 or 2,6 linkage, which we were interested in. And if you do that on fetuin, which contains both, so that's the second panel here, you see clearly two peaks for the sialyl lactose. And we assigned them as alpha and beta, and we could assign the left-hand one as the 2,6, the yellow one as the 2,6, because we had a standard that we knew only contained the 2,6 ranks. So then if you treat this with an enzyme called NAN-B, which is a 2,3 sialidase, you get the yellow because you cleave the 2,3. And with our pseudosialidase, you can see you get the other peak. So in this very complex protein context, we could actually show with this method that we get really good selectivity for the 2,6 sialic acid linkage. We've also collaborated, tried to introduce this method into other workflows. And you all know the beautiful work Pauline Rudd's been doing, and she has this lab in Singapore. And I was very lucky to have a joint student with them, A-star Manchester student, Ed Pallister, who was a really very talented student. So this is him in Singapore. And we published some really nice papers adding ion mobility to the HILIC protocol. So Alistair's project was glycosylation of alpha antitrypsin. So alpha antitrypsin is a biopharmaceutical. Silylation is extremely important. From their fermentation procedures that didn't get very high silylation. You can see here the silylated form has a circulatory half-life of 5 days, desilylate of only 5 minutes. So really, we wanted to increase the silylation by doing silylation using silyl transferases. So that was sort of his starter project. And he did that with a bacterial silyl transferase, which we could express in E. coli. It's quite a cheap process, really. CMP NANA, we can regenerate, again, using bacterial regeneration enzymes. So that worked really quite well. But because, they wanted to analyze the glycans, he did a full HILIC analysis on the product. And to his surprise, he found a structure that seemed to be over-silylated. So when he looked at that terminal silyl lactose using sort of similar approaches I've shown before, he clearly found that there was a motif that had the sort of the silyl lactose, and the silyl lactose had a further silylation. And this was this is an extracted ion chromatogram on different glycosylation sites, and he saw this structure clearly on different glycosylation sites. So there seemed to have been some over-silylation happening, and we wanted to know which one that was. And you can imagine that's quite difficult to determine on something like complex like a glycoprotein. So we had sort of hypotheses that it could be one of those 3 structures. The left-hand one, I think we've just seen this previously, but it didn't really fit with the substrate specificity of our enzyme. But it could be that one that has this sort of internal extrasilylation. It could be a disilylation, but then you needed 2,8 silyl transferase, so that was also a little bit unlikely that our enzyme would do that. And then we found in the literature one report of a structure, only a free oligosaccharide, which had this interesting 2,3 plus 2,6 silylation. And so we were wondering which one of those it was. And after quite a long story, he could show that with this ion mobility fragmentation approach, using a standard of all of these, that the best fit was clearly with this unusual disilylated structure. So the enzyme was putting both a 2,3 and a 2,6 sialic acid onto this terminal galactose, which is obviously very relevant when you remodel glycans, that you know exactly what you have in your mixtures. They then extended this to sort of address some long-standing issues of heterogeneity with these HILIC profiles. I mean, we've seen a lot of these HILIC profiles. I think Pauline has really pioneered these. And really, there's still quite a lot of ambiguities. This goes back to this sort of sharpness of this image, as you all know. If you look at assignment of the peaks, people don't know really which arm is glycosylated, which arm carries which glycan. As I said, do you have 2,6 or 2,3 sialic acids on some of these? So there's still, despite the fact that you have so many peaks and you've got this sharpness, there's still some ambiguity. It might not matter in every experiment. Maybe with high throughput experiment it doesn't matter. But we should have methods to resolve this. And by taking these peaks and then sending them through an ion mobility, so basically sticking with their method, their HILIC method, but just adding the ion mobility step to it in line, they were managed to actually really separate a lot of these isomers. Because it's a fragment-based approach, it doesn't really matter whether you do glycoproteomics or whether you use these labeling techniques. Because we're only looking at these fragments, it shouldn't really matter where they originate from. And Ed did it both, and really wanted to resolve these sort of isomers. As I said, the alpha 1,6 and 3 arm, of the core. various isomerizations. And really, he published a very extensive analytical chemistry paper, just to show you just one set of data. For example, he wanted to know whether the galactose was on the 3 or the 6 arm here. So these are two clearly isomeric fragments, which are difficult to separate by Hillig. And by fragmentation, you could look at fragments that are very diagnostic, depending on, you can see these tetrasaccharides really are different depending on which arm they're on. Obviously, you can ignore all the other M over Z fragments, so you send them through the ion mobility tube, and you can see, first of all, regardless of whether you start with labeled glycans that have PNGAs released or whether you start with glycopeptides, you really get very reproducibly exactly the same profiles. So really, again, emphasizing this technique is really quite reproducible. And then when he had it on the 3 arm, you could see he get very reproducibly this extra peak at 2, 3, 4. Again, regardless of the analyte. So you can distinguish between those. And in this paper, there are many, many more experiments on other isomers, which I won't talk about. And I just want to finish with the fact to say that other groups now using this. There's a very nice paper from Rebecca Miller and the Bagel group where they did this for GAGs. So they wanted to just know whether in this very large GAG there was that pentasaccharide or hexasaccharide sequence, whether it was there. And by using standards, you can see here in C, A, B, C, D, substandards, which are much more easy to access synthetically, and then comparing the ion mobility fragments and so on, they could actually assign the sequence really unambiguously and show that that particular sequence was also there. So this approach doesn't only work for N-glycans and metabolites, it also works for GAGs. And I think it's a very, very nice method if you want to get very, very high resolution of structure determination, which in some cases might be important. So really, I think we have now 2 techniques, really. The top technique is perhaps more amenable if you want to do glycomics experiment. You label, you get the full fragments, and then you separate them by LC methods and so on, and compare them to standards. So I think we've had a lot of very beautiful examples here. So it requires really big databases. That's a sort of one problem, and large number of standards. But of course, the data are much more simple to interpret. Claire Ayers and Perdita Barron, Chris Gray and Ed Pallister have really been the PhD students in my group who have really pushed this. So I have to really thank them, thank my collaborators, funders, and I thank you for your attention.